Welcome to my introduction video for UCUR 2022. My name is Yongqi Lan. Thank you for being here today. I'm a student at the University of Utah, and I'm glad to share my project, which aims to unravel the role of the protein Slm1 in mitochondrial function. To understand Slm1, we must first begin with water. Water is a common environmental condition that each cell would encounter in nature. When a rainstorm hits, an yeast cell could be flushed away from its original environment, such as a fruit, into pure water. To respond to the increased tension on the plasma membrane caused by water, furrows on the plasma membrane, isosomes, would flatten. In the meantime, one of its protein members, Slm1, would leave isosomes and relocalize to a protein complex called TORC2, which then upregulates sphingolipid synthesis to relieve the membrane stress. So far, this is what is known about Slm1. However, there is more than just tension in the combo of shocks induced by water. There are also starvation, ion depletion, and many other stresses. To respond to starvation, for example, will require some regulation on the overall metabolism, have something to do with the mitochondria. And this is where my research project comes into play. To understand the project, we must first define the segments of Slm1 using the research. The N-terminus of Slm1 is our region of interest. It is a region containing most of the protein sites for regulation, as well as potential sites for protein phase separation. The remainder of Slm1 is defined as the C-terminus here, just for convenience. The three versions of Slm1, N-terminus, full-length, and C-terminus, are each tagged with green fluorescence protein, GFP, then transformed into E cells for imaging. The key finding is that Slm1 localizes to mitochondria when the cell is moved from standard condition into water, causing mitochondrial fragmentation. Following the finding, we further identified the N-terminus of Slm1 to be critical for mitochondrial fragmentation. Slm2, the parallel of Slm1, is also essential to cause the phenotype. In the latest experiments, we find out the two exact factors in the combo shocks of water that causes fragmentation, which happens to be a combo of hypoosmotic stress and starvation. Overall, despite the exact physiological mechanism behind the story is still under investigation, the research reveals Slm1's relationship with the mitochondria, making what was thought to be a plasma membrane protein to metabolism. This opens a new chapter to a potential pathway we have never thought of. This is the end of my introduction. Thank you for listening.